Okay, in this video we're going to learn how to subculture HaCaT cells. So the subculturing cells, also known as passaging or splitting cells, you may hear all three terms used by different people. They all mean the same thing. Basically, we've got cells that are fully confluent on the plate, and we need to subculture them. So we want to basically, we're going to use an enzyme called trypsin to detach the cells from the plate and then place a sub-fraction of those cells onto a new plate that will allow them to continue to grow. And we do this to maintain cells in a healthy proliferative state. Some cell lines are not very happy if they're confluent and touching each other, so to keep cells growing well in a healthy way, this is important to do. Okay, so first thing we want to do is make sure everything is ready. So one thing I've got done, or I've already started, is to warm up the media and trypsin. So we have both, for these cells, we're going to use DMEM, where we've added fetal bovine serum and pen-strep L-glutamine. So penicillin and streptomycin are antibiotics. L-glutamine is another essential amino acid, so sometimes that gets added in an extra amount to media. We also have trypsin. In this case, it's trypsin 0.25% that's in a 50 ml conical bottle. We also have trypsin, maybe not in some in this freezer here, that's in a 100 ml bottle, which can also be used. So there's both trypsin and EDTA. EDTA is a compound that will chelate divalent cations, and that also helps to detach cells from the plate. So a good first thing to do is kind of wipe down surfaces. We don't know who was here before, we don't know what they've touched, we don't know what bacteria they're carrying around, so anything you're going to touch with your gloved hands can also be good to kind of wipe down with some ethanol and help kind of keep it in a sterile, as sterile as possible surface. The UV light is on there to help sterilize the surface from the previous user. So all we need to do to get this set is open up the sash here up until about that level indicated there. And we'll turn the light on with this button up here. You can see there. And now the light's on. Okay. One thing we could do, and I've got a protocol in the lab handbook for doing this. If you want detailed protocols to look at while you're doing this, you can just tape it up above the hood there. So got the taped up there. Okay. So now we want to get everything in the incubator to be ready to trypsinize the cells. So I'm going to take the media and the trypsin, get rid of any excess water here, and then what we're going to do is we're going to bring it over into the hood. But before we put it in the hood, we want to spray everything down with ethanol. So use plenty of ethanol, not a problem. You can always make more. Some people wipe it down. It's good actually to have the ethanol sit on there for a little bit to help basically dehydrate and kill anything that might be in the water bath, for example. And then we're going to set everything up in the hood. One good thing to get in the habit of, so you don't have to unloosen everything while you're handling something else, is to maybe loosen the lids, not take it all the way off. Again, if things are off, bacteria, fungi, whatever can fall into your solution state. Another thing we're going to need is PBS to wash the cells. So it's phosphate buffered saline. We'll also spray that down. Okay. So those are the three things we need to start to split the cells. So I'm going to get the cells out of the incubator. Always a good idea on a daily basis to check your cells, see how they're doing. If you have a lot of cells floating or other things floating in the media, that's a good sign that something is wrong, which is not a good sign. And so the cells look pretty confluent. I doubt that will be visible here. Nope, but I thought I would try. Okay. Anyway, you'll get used to it. If the cells are fully confluent, there won't be any empty space on the plate. Now some cells are not happy if being too confluent, so you really have to know sort of the personality of the cell line you're growing, and every cell line has its unique properties, and you just have to get used to those properties. Okay, so the first thing we want to do is aspirate the existing media. So turn the vacuum on over here, turn it counterclockwise. You kind of hear vacuum air coming up. I'm going to take one of these glass pipettes here in this metal box. Put it on the end of the tubing here, put the lid back on again. You always want everything covered as much as possible. And then what I'm going to do is just basically tilt the plate a little bit, stick the glass pipette to the bottom corner, one of the corners of the, one of the ends of the plate, and you'll see the media aspirate. And then what I'm just going to do is just kind of hang this up here so I can use it in a minute. Now, next thing we want to do is wash with PBS. And so I typically use 2 mils. If you're new at this, and anywhere from 2 to 5 mils is probably fine. And so we can use either a 5 mil pipette or a 2 mil, the green ones. I typically use the 2 mils just out of habit. We'll use 5 mils today just to, for new people learning how to pipette. Sometimes it's easier to use because it's a larger, it can take a larger volume and you're less likely to pipette too much. So what we're going to do is attach this to the end of the pipetter here, and you always want to take put the paper back in the back, plastic in the front, break it towards you, and that'll break the plastic, so break the paper in the back, stick it in the, with a reasonably firm, so now it looks like it's pretty secure in the pipetter. We'll throw that away in the trash. Importantly, you always want to have everything in the back in the hood, so you don't want to take this outside of the hood, or else now it's potentially contaminated. So what I'm going to do is pull up 5 mils of PBS. It doesn't need to be perfectly exact. It looks like this is probably closer to 5.2 mils almost. Exact amounts is not too important. The key thing is we want to wash the cells. So we're going to add this enzyme trypsin in a minute. Trypsin basically degrades proteins on the cell surface that allow the cells to attach to the plastic. And one of the proteins, or the major proteins in media, there's a variety of growth factors and other things, but again, the growth factors come from this fetal bovine serum. Albumin is a major protein in blood, and so trypsin will degrade lots of proteins, but if it's covered, the cells are essentially covered with albumin, and so what we want to do is wash the cells so that the trypsin can, the albumin is washed away as much as possible, and trypsin can act on any proteins on the cell surface. So then we're going to aspirate the media. And then we're going to add 2 mils of trypsin. Again, this could be used either with a 5 mil tube, or 5 mil pipette, sorry. You can go anywhere from 1 to maybe 7.5 mils or so. I'm going to use the green 2 mil pipette here. You always want to get used to looking at what the numbers and what the breakdown is, the gradations on the pipette tips. So I'm going to pull up 2. You can see basically the way it's numbered. If you look closely, you can see 1.8 all the way up to 1.0 and then 0. And so if we go all the way up to 0, that's going to be 2 mils. And then I'm going to add this to plate. And anytime you have cells that you've washed in PBS, you don't want to keep them there too long because eventually the cells will dry out. So I'm going to shake this back and forth a few times and and then what we're going to do is put the cells back in the incubator. Trypsin is an enzyme, and like many enzymes, they have an optimal temperature at which they act. So trypsin works quite well at 37 degrees. And so the trypsinization process to get the cells off the plate will work much better when the cells in the incubator. Now while we're waiting, we can get our plate ready. So again, we're going to be self-culturing the cells to continue to grow them. I'm also going to plate some cells for an experiment while I'm trypsinizing. That's typically what you're doing. You may be splitting your cells to plate some cells for an experiment and then allow some to continue to grow so that you can maybe do another experiment in a few days. I'll take this up. You can, hopefully these are open the right way. So the way they should be opened is torn from the top down, which means the plates are upside down, meaning the bottom plate is upside down relative to the lid. And when we're done, we're going to wrap that back up, tape it shut, because we don't want to contaminate anything inside the cells there, inside the tube of plates. And I'm going to grab some cells to plates, and grab some additional plates to place some cells into a 24-well plate for an experiment. And so while we're waiting for the cells to trypsinize, we can get everything ready so we can label the plate. So these are HaCaT cells. I believe this is passage 70. I'm going to put my initials so it's not my plate, it's somebody else's. And then put the date. So today's September 21st. now that we're done with the PBS, we can actually put the trypsin, we can put that away. So again, it's going to take 5 to 10 minutes for the cells to come off the plate. So while we're waiting and get things ready, I'm going to go put that PBS away there. Put the trypsin back in the refrigerator. Located down there. And so again, like I said, I'm going to plate some cells into that 10 centimeter or 100 millimeter plate. I'm also going to plate some cells into a 224 well plates. And it looks like we're out of 50 mil conical tubes. So I'm going to go over get some in the other part of the lab. Good thing to note is always have everything stocked as much as possible. So better thing to do would be for me to have checked that I had what I needed before I started this process. And always a good idea to be prepared for what you need. Just in case something is really time sensitive, you need to hurry up, get something done. In this case, we have a few minutes, so it's not really too important. Okay. Okay, so I'm going to put this in here. And so now while we're waiting, I'm going to get the media, put them in into the plate, into the 50 mil conical tube. And anytime I've gone out and touched something else outside the hood, I'm spray my gloves down with ethanol. Don't trust the sterility of any surface outside the hood because it's not sterile. And even within the hood, it's not totally sterile. It's we have a wall of air here that helps to protect, limit what gets in and out of the hood. So I'm going to take it for these 10 centimeter or 100 millimeter plates. Typically use 10 mils of media. We'll use the orange 10 mil pipette. I'll pull up 10 mils. Put that into the plate. So that's me for subculturing the cells. I'm also, like I said, plate some cells into that 224 well plates. So I'm going to take, this is not in this particular protocol necessarily, but I'm going to add 24 mils of media. So 12 times 2. Again, this is a 10 mil pipette, or called the 10 mil pipette. So you can see it goes up to, I'm not sure how clear it is here, but it goes up to 10, but you can still see there's additional volume above it. So you can actually go up to, so this is 10, minus 1 would be 11, 12, 13 mils, pretty accurately, or very accurately. Okay, so now that we have everything ready, we can go double check our cells. Spray my hands down again, just out of habit. The more you do cell culture, hopefully the more you get used to spraying your hands down as much as possible. Okay, so typically it's hard to, after just a few minutes in there, you might not be able to see much in terms of cells coming off the plate, so you need to use the microscope to take a look. I'm going to take a look at the cells here, and what you'll see if you look at the microscope, and again I can't really show this very well with this particular microscope, is now the cells have started to round up, whereas they were spread out, covering the plate. Now you can see the cells are definitely all round, and they're now spaced between some of the cells. And so that, not quite ready. One thing we could do to kind of speed things along is just kind of actually hit the plate, kind of tap the side, and so that can be useful to see. Sometimes that helps to dislodge cells, and it actually looks like some of the cells are starting to come off, but we're not really done yet. We want all the cells to come off, so I'm going to put it back into the incubator. And that's important, depending on what you're doing. You want to be sure the approximate number of cells you're plating. Again, we're not going to count any cells today. That's a separate protocol we can show how to do. But once the cells reach confluence, there's some range at which the cell number will be, and so it's probably typically around 10 to 12 million cells, at least for this particular cell type, can fit in a confluent plate of cells. And so again, if you trypsinize all the cells, you have 10 to 12 million cells in suspension. We can take a fraction of those cells into a new plate and have a general estimate of how many cells we put in a new plate. Now, if only half the cells come off the plate, now we have a different number. So it's very important to have some idea of what percent of the cells came off the plate and how many you're plating. If the cells, and so again, the trypsin is going to allow that to come off. We don't want the cells to sit in trypsin for too long. Again, trypsin is degrading proteins on the cell surface. Cells are, if you have cells in trypsin for hours, the cells are eventually going to die. And so That's why what we're going to do is basically stop the trypsinization by adding full media. And as I mentioned earlier, the media contains fetal bovine serum. A major protein in fetal bovine serum is albumin. So that essentially, to some extent, neutralizes the trypsin. So it basically stops the trypsin from being able to act on the cell surface, because now you have all this other albumin for the trypsin to act on. Now the enzyme essentially will, be done doing its job. There's not much trypsin in here relative to the amount of albumin. Okay, I'm gonna go check again in another minute or so. Okay, one thing you can do too sometimes is this is not 100% perfectly level. Sometimes you might notice there might be a slight tilt to the plate. So sometimes after I have it in there for a few minutes, I may turn the plate 90 degrees or so just to make sure everything is fully covered. And the more you go in there, nothing wrong with checking your cells. Okay, so really hitting it, and it's hard to see without looking at it. Sometimes you actually look at the light and start to see the cells. Look at the light. It's kind of weird to show you, but you can see cells now really coming off the plate. And now if you look at the microscope, all the cells are floating. Sometimes clumps of cells, but they're all fully dislodged from the plate. Spray my hands down again. And now what we're going to do is add media. I think the protocol says 6 mils. I typically do 5 or 6. Part of this is sort of eyeballing it and understand the density. These HaCaT cells are pretty strong cells. They don't mind going extra days at full confluence. They can go a long time at full confluence, actually. And so sometimes you can look at cells look like they're fully confluent. The next day, they may look like there's more cells because they actually kind of shrink up a little bit. So I'm going to go ahead and pull up 6 mils. And then I'm going to pipette it onto the plate. And if there's any area, so you can kind of see, you'd have to look yourself at the cells. There may be regions on the plate where those look like there's some cells still attached. What you can do is pipette up and down and dislodge the remaining cells. So I'll go around the plate a couple times just to make sure I've dislodged all the cells. And what I'm going to do is pull up 2 mils of media and then add that to this 10 centimeter plate. And so again, we had 2 mils of trypsin, 6 mils of media. That's a total of 8 mils. I've taken 2, so 1/4 of that and do a new plate. Cells grow, estimate they double every 24 hours or so. Probably 2, sometimes 3 days. And there's some recovery period after plating them, so they don't just grow immediately. It takes a few hours for the cells to attach to the plate and then get into a state where they can grow and proliferate again. But within 2 or 3 days, this plate of cells will be confluent. And then I'm going to take a little over 3.5 mils into this conical tube of plate. And so this we're done with. So what I'm going to do is I'm going to take that plate and put it back into the incubator. And what we'll do is, again, every day, it's always a good idea to check your cells every day. See how they're growing. You can see the kind of health of the cells by looking at that. And then once we're done with this plate of cells, what I'm going to do is aspirate the remaining cells and put this into the biohazard under the hood. So again, not part of the particular protocol of subculturing cells here is to plate some cells. So I'm going to do an experiment. So again, typically I don't just split cells. Typically I'm splitting cells to continue to grow some and also splitting some to do an experiment with. So in this case, I've got 24 mils of media that I added 3.5 mils of trypsinized media, trypsinized cells to. I'll put this in this boat here. Again, this is a bit of a sterility risk. Again, it's not covered, but that's we're going to use this multi-channel pipetter to pipette cells into these 24 plates pretty quickly. But again, as a general recommendation, you don't want any solution of cells or media just lying around without being covered. Pipette, now to resuspend. The nice thing about this multi-channel pipetter is it allows you to pipette 6 wells at one time. away. Biohazard. Aspirate that. Stick the cells into the incubator. And then we're done. We're going to clean up. So I'm going to take the media, bring that back into the fridge. And then what we want to do is clean up, sterilize, or clean out the tubing. So again, all of that waste is going to attach to that glass pipette. The tubing goes into this waste container over here. It eventually gets filled up, and so we'll need to empty that when it's full. But that usually takes maybe every couple of weeks or so. But to prevent anything from growing in the tubes, what we're going to do is we're going to got some 50% bleach. So run a bit of bleach into there until you start to see it coming through to the end where you can see it going into the outside the hood on the right wall of the hood. And then this is some 70% ethanol. So I usually do bleach and then ethanol. Put the lid back. Put it back over here. Recharge, make sure this plugged in so it's always charged. If you spilled anything, it's always a good habit to get into spraying all the surfaces down with ethanol. Take a Kim wipe, wipe things down, and then we can close the sash. And if you want to be also good, you can clean anything you've touched here. So again, as clean as possible. And then last, what we want to do is turn on the UV light. So we're going to hit the UV light there. That'll turn the white light off and turns on the UV light to sterilize all the surfaces in the hood. And so that's how we subculture HaCaT cells.